Hi, do you see the screen?
Yes.
All right. So hi, I'm a third-year QLS student, and today I'll try to take you through a way of doing epigenomics that does not ignore the genetic variation that might be present in the population that we're studying. So first, I'll try to kind of explain to you the utility of epigenomics. So the reason we do epigenomics is because it's a key tool of understanding the function of genomes. So we use methods such as CheapSeq and ATAC-seq to reveal the transcription factor binding sites and modify the histones, and also to identify areas where the chromatin is accessible or not accessible. And all this data that we generate plays an important role in mapping causal variants and fine mapping variants that are responsible for a certain trait. So for example, here I have an Alzheimer whole genome-wide association study. So in this study, they identified a cluster of SNPs that are together associated with the trait because of linkage disequilibrium. And the main task is to kind of prioritize these SNPs and identify one that is likely the causal variant. And here in red, we see that one of these SNPs is prioritized because it's in a region of the genome that is accessible, where the chromatin is accessible in the cell type that is relevant to the trait. And it also modifies the transcription factor binding motif that we obtain with the transcription factor binding methods. And all these evidences come together to tell us that this SNP is more likely to be responsible for the trait than the surrounding motifs, the surrounding SNPs. So the way we generate this data is really from until now is really a short read sequencing experiments that we then map to reference genomes. And then you will see that when we talk about reference genomes, we are introducing a source of bias called a reference bias. And also because the sample we study, the genotype is not identical to the reference genome, we also lose some allelic specific information that could be useful in understanding the trait more. So just to tell you how bad allelic specific allelic bias and the reference bias is in our current approaches, here I am showing you the partitioning of signal in a heterozygous polymorphic loci, right? So we expect that the signal to be equally distributed between the reference allele and the non-reference allele. And indeed, for small variants, we do find that this is the case. We find the proper proportion of signal on both alleles. But as the variant increases in size, so as we approach indels above the 40 base pair threshold, we lose more and more reads that should belong to the alternative allele, right? So this problem is sufficiently significant that some approaches were already developed to address this, pipelines like allele-seq on WASP. But one shortcoming is that they do not model the relationship between genomes in a population. So it's difficult to scale to multiple genomes. So one of the tools we could use to scale this to multiple genomes is through the concept of the pangenome, here implemented as a genome graph. So what a genome graph does is takes a large genomic sequence and it splits it into pieces, and these pieces are called nodes. And then we reorder them and relay them through edges so we can reconstruct the original sequence. And here, if you see the example of the HLA locus, which is known to be quite diverse in humans, we see linear parts where there is no genetic variation, and then we see polymorphic parts that are indeed variable among humans. And the genome of each individual would be a trajectory that starts from this end and crosses over, taking a certain path to the other end. And if you prefer a more linear representation, here we have a linear track that kind of tells you the absence and presence of various alleles at every point in this in the HLA locus, right? So if you could build such a representation for a large number of genomes, we could do two things. First, we could correct reference bias in epigenomic studies, and also we could obtain allelic specific information on the epigenomic signal that we encounter. So I tried to do just that, and by applying this the concept of the genome for 35 genomes, that were studied in two conditions, an influenza infected condition and a non-infected condition. So in this way, we have some biological context that we will use to make the data more interesting. And we will use this fact to identify some interesting causal variants, some expression QTLs later on. So as you can see, this data set is quite rich. Each individual is sequenced using linked read libraries. And also each individual basically donated macrophages that were influenza infected or naive, and were sequenced under these two conditions with ataxic, RNA-seq, and ChIP-seq. So we can do quite a lot with this data. So the first step was to use the linked read the whole genome sequencing data to call variants, indels and SNPs, and construct the so-called pangenome graph that represents and relates all these genomes together. So as you can see, I recovered around 3 million insertions and deletions for each genome, and sorry, not 3 million, 300,000 insertions and deletions per sample, and about 4 million SNVs per sample, right? And we found that by using a more representative genome reference to map epigenomic signal, we can improve read mapping and rescue more unmapped reads that would normally go to waste. And again, we found that downstream it has an impact. So by using a more representative genome reference, we detect more epigenomic events than as before. So for each individual, on average, we identify 2,000 to 6,000 new epigenomic peaks or events per sample. And of course, we wanted to know if these new epigenomic peaks replicate well across the cohort and are not just very rare singletons. And through a permutation experiment, we concluded that indeed these new peaks, new events, they replicate better than expected by chance, right? So this is evidence that this is not just like a different sort of noise, but indeed it's a true signal. And of course, we don't want to limit ourselves to just indels and SNVs. The main reason we're pursuing this kind of representation is really to apply it to structural variation, which other studies show that they are leading causal variants in many cases, and could explain some amount of missing heritability, because we're not very good at working with structural variants at this point. And some of the inspiration we had for this is really another study that brings in transposable elements in infection, right? So Cedric Fechot and his lab found that some transposable elements carry transcription factor binding motifs that participate in immunity. So we kind of copied this hypothesis and wondered if another class of transposable element polymorphisms, ALUs, could also play a role in influenza. And we developed a special pipeline to kind of retrieve these structural variants, resolve their sequence, and genotype every sample and construct, again, the genome graph that represents these polymorphic loci. So after going through all this exercise, in the end, we have the structure that has in gray the reference path, and in red the insertion polymorphism that we recovered. And using this, we will recover allele-specific information that belongs to this insertion that is about 300 base pairs long, and also recover lost reads that should be mapping on this insertion but are normally lost. So among the 35 individuals, we identified about 9,000 mobile element insertions, ALUs, and we successfully recovered about 70% of these events, so about 5,000 events. And again, we think we did a good job because it passes some checks. So once we recapitulate these reassembled insertions, we recovered the original population structure that we know is in the data set. And some of these events are quite interesting without even further analysis. For example, some of these insertions are within exons, and another set of these insertions are within enhancers. Now, of course, we only study one of these cell types, macrophages, so not all these events could be relevant because they are expressed in other cell types, but still we might see a subset of these. in our analysis when we go on. So because I wanted to make sure that I'm doing a good job, and because we know that these sequences are repeats, so there are many copies of these sequences across the genome. I want to make sure that when I'm mapping reads, I do it with sufficient specificity, and I also don't lose any sensitivity. So the way to do this was basically through a regenotyping experiment, where I used this reference pangenome that I developed to realign whole genome sequencing reads and regenotype every locus and see if I am losing genotypes or I'm getting too many genotypes, right? So that would, one case would point to a lack of sensitivity, and the other case would point to a lack of specificity. So I found that actually I have pretty good sensitivity and specificity, so I'm mapping reads correctly. If you're interested to know how a mapping looks on one of these loci, I have an example here. So again, we have the red part, which is the insertion allele, and the black part, which is the reference allele, right? And genomes that have the reference allele, they go straight through. They take a direct path, and the genomes that have the insertion allele, they take the detour. They follow the black segment. They go through the red line and then proceed on to the rest of the genome. So when I look at an epigenomic signal, so H2K4 monomethylation in this case, which is a histomodification, I find that I do a good job in retrieving allele specific signal. So on the y-axis here, I have reads that were assigned to the insertion allele in these heterozygous polymorphic loci, and the x-axis describes reads assigned to the reference allele, right? So the green points indicate to epigenomic events that are wholly supported by the insertion allele, and the red points indicate to epigenomic events that are equally supported by the reference and the insertion allele, and the blue events are just supported by the reference allele. So all these are interesting, but using this sort of approach, we can have a population scale map of these events. So we can tell which locus and which sample supports a peak on the insertion, right? And because all the genomes are related together in a pangenome, we don't have to do expensive obscure operations of kind of merging the data together, because the genomes are already merged together, right? So just to show you the most interesting example I found in this cohort, using the RNA-seq data, and again, using the ATAC-seq and the ChIP-seq data, we were able to find a mobile element insertion. So in this case, an allo YH3 that is associated with the expression of TRIM25 gene, which is a gene associated with repressing influenza virus transcription, right? And we found that this polymorphic locus supports HUK27AC acetylation, HUK4 monomethylation, and is an accessible chromatin in about 29 of the 35 individuals that we have in the cohort. So again, this is a data point that would be difficult to retrieve with just a regular reference that only represents a single genome, because again, the insertion would not be represented there, and we would not be able to retrieve the signal very easily. So I'm going to finish here. So really the strength of this approach is that we can represent structural variation in many genomes and map epigenomic and transcriptomic signals to structural variant alleles. And this will allow to apply fan mapping techniques to structural variants at the scale of populations. So I hope you found this interesting, and I'll take questions now. Thank you.
Thank you very much, Cristian. Again, a quick question for Cristian, or a couple of very quick questions for Cristian. Does some of this work relate to the, oh, yes, you still have 15 minutes, Jeffrey, don't worry, we'll run a little bit over, but one quick question. Does some of the work you're doing relate to the evolutionary stuff that Mathieu showed in his keynote talk at the beginning of the day, Cristian?
Yeah, well, we do have some like a evolutionary perspective. We think that some of these transposable element families carry transcription factor binding motifs, and as they propagate through the genome, they lead to biological innovations, right?
Okay, I think we'll move on. We will now have our last speaker.